Good afternoon, and welcome back to the third session of today, Optimizing Contamination Control. We've got a great panel ahead of us. My name is Ian Betteridge. I'll be the moderator for today. I'm Head of Content for European Pharmaceutical Review. And just to give a quick introduction to our three wonderful panelists, we have Hayden Skalski, Life Sciences Product Applications Specialist at Veolia. Mark Holworth, Global Pharma and Senior GMP Advisor for Particle Measuring Systems, and Vanessa Vasadi Figueroa, Sterility Assurance and Contamination Control Leader for VVF Science. I just want to start with a little bit of an open question for each of you. And contamination control sits at the intersection of microbiology, engineering, regulatory compliance, and operational discipline. It's really central to kind of everything that we do in pharma. We've got different perspectives and we'll do different systems. I'm just going to turn that off. Somebody trying to get hold of me. We've all got different perspectives. So before we get into the substance, and maybe you can answer this one first, Mark, and we'll turn to the others in a sec. But what does the contamination control problem look like from where you sit?
Thanks for that. I think there's a sense of the systems are drowning in data, and there's so much data being generated, and the workload. When you look at a typical facility, 2,000 samples, 2,000 microbial samples per week, a similar amount of particle count data per week. There's the automated side of things, and there's so much data coming in to a small team. Having the bandwidth to actually analyze it and decipher what it's telling them about the environment, and employing that understanding is kind of the hardest thing right now. Having availability of technology is fairly straightforward, but understanding what it's telling you, that's the trick. And the trick is time. Nobody's got the time to do that analysis now.
I think that's a really good point. And my background is kind of specifically in data and technology. So there's an old phrase that we use in computing a lot, which is garbage in, garbage out. And sifting out the garbage is always one of the hardest challenges that you've got. So Mark, over to you next. What would you say is kind of, how does the contamination control problem look from your perspective?
I think you meant Hayden or Vanessa, because you've got me, Ian.
Oh, sorry. Yes, Hayden.
I understood that as Hayden too in my head.
Very strange. Hayden, we'll come to you next.
I can go by Mark as well. Thanks. Yeah, good question. So from where I sit, which is the microbiology world, a contamination problem often doesn't announce itself with obvious failures. It's the subtle things, I believe. It looks like creeping upward trends in environmental monitoring data, unexpected endotoxin spikes in WFI systems after a maintenance or a shutdown, inconsistent spike recoveries in your traditional microbiology QC testing, or even rising in-process bioburden that hints at maybe resilient gram-positive or gram-negative organisms, or potentially even some biofilms that are hiding in the background of your water systems. I would say it's those early signals that traditional bioburden counts might miss, but a strong contamination control strategy with layered monitoring and rapid micro methods can catch proactively. So, my role in this industry and in my current role of my career is, turning those signals into actionable improvements before they reach the product for patient safety.
Great stuff. Vanessa, over to you.
Yeah, I think you can sense based on Mark and Hayden's response that the people looking at the systems and interpreting the data need to have the credentials. And, contamination control has become so wildly popular that it's almost common language for every single person in the company. And this is actually both, a good thing, but also concerning because contamination control is not just interpreting data. Contamination control is bringing context to that data based on what you know about the process, about the product, about the, real boots on the ground experience. And it's a tricky thing. Microbes are tricky, right? Understanding them in context or based on the specific product or process you have is so much more important than the data itself. Because you don't want to overreact and you don't want to put in controls that don't bring value. And you don't want to underreact and sort of say, oh, it's isolated, which is what we're known to do in our industry, when that isolated incident could be a window into a bigger picture, like a biofilm or something like that, right? And so I think as we sort of take a breath from this wild activity over the past few years trying to implement EU Annex 1, we need to make sure that we're always considering the context, that we understand the way microbes behave, and that we put that in perspective of what we're doing at the moment so that we don't overreact to, right? It's easy when you're manufacturing sterile products because it's sort of a black and white situation.
Mm.
But that's when it comes to finished product testing. The environment, understanding in-process controls, those are all really critical and they need to be right-sized. So I see a lot of us needing to course correct and to take a breath and to make sure that we are applying the right controls at the right point in the process. And so hoping, yeah, we'll talk more about that today.
Definitely. And in fact, that kind of leads me on to the first section and the first topic area that I want to touch on nicely, because this is a constantly evolving landscape. And from my perspective, and correct me if I'm wrong, it almost feels like contamination control is actually harder now than it used to be. You always hope it's going to get simpler, but that doesn't seem to be the case. You've got, as you mentioned, revised Annex 1. You've got the expansion of sterile manufacturing into biologics, cell and gene therapies, other advanced modalities. And that feels like it's kind of raised the bar for what contamination control needs to achieve. Vanessa, maybe this is one for you. So Annex 1 set kind of new expectations when it came into force, and the range of sterile manufacturing modalities keeps expanding. So is it true that the bar for contamination control is actually higher, or am I just misreading it?
You're not misreading it. And I often talk about this because when people are sort of trying to scramble with pulling together a meaningful contamination control strategy, people are like, their perception is that this is wholly new. Yes, the bar is fundamentally higher today than perhaps it was 5 years ago or 10 years ago. But that's not necessarily because the science of contamination has changed. It hasn't. It hasn't at all. The industry's expectations around understanding contamination control, controlling contamination, and then documenting it because we are in a structured quality system compliance industry, those have evolved significantly, right? What Annex 1 did is they formalized in a very um, in a very structured way about 12 to 15 different elements that they're saying from a regulatory perspective you must have described, and you must tell us your position on how you plan to control all these different aspects of contamination, right? And there's detection, there's understanding prevention, and then there's understanding risk mitigation. So it's not just contamination control, it's all the ways in which you're going to prevent it from entering your process, how you're going to reduce it if you have it in the product, the process, the API, etc. And then how you're going to detect it if those controls don't work or the control is right size based on the type of product, right? So if you look at the 2004 FDA guidance, contamination control was an expectation.
Mm.
I have been developing contamination control strategies. We call them microbial control strategies in the past for the entire duration of my career. So they're not new to me. They're not new to those who do contamination control, but they have been sort of become they've broken open into the larger mainstream of pharmaceutical quality system management, right? So 5 years ago, perhaps organizations viewed contamination control as a collection of separate different programs, right? Even sterility assurance was the beginning of what we call this collection of different contamination controls. Today, regulators are expecting all of those separate systems to be interconnected. And that's the key, I think. That's the biggest shift that we have, that we go from compliance-based thinking that it's a checkbox. I've got the program in place, I've got EM, and it looks really good, as to a knowledge-based thinking. Manufacturers are now like expected to not only have the controls in place, but understand why they're there, how you can shift the contamination controls more upstream to be preventive, and whether or not those controls are effective and how they interact, right? So I think that this is mirroring the complexity that we're also seeing in technology, in manufacturing systems as manufacturers are bringing on isolators, closed wraps. We see gloveless robotic filling systems. We have a lot of single-use technologies, which is great from a sterile control perspective. But you mentioned, Ian, you mentioned cell and gene therapies, right? And personalized medicine. So we're seeing both complexity in the technology we use to manufacture, but also the types of products that we're manufacturing. So it just makes sense. Lock and step with that complexity are the expectations around contamination control. So perhaps to go full circle, yes, the bar is higher. The formality and the way that we understand contamination control is expanding, and the expectations are deeper than they had been in the past. And so I think from my perspective as a microbiologist and sterility assurance professional, it's great. But the question now we're going to settle back into is, how are people doing this? How well are we articulate the way that we're controlling contamination? Now people are going to be judged on that in regulatory inspections. And that's a whole mixed bag of how well people are doing that, right? How well we're understanding the science of contamination control. And so, it's just a matter of time before we start seeing all different versions of CCS documents and the levels of maturity associated with them. So, yeah, but it's an exciting time.
It's definitely an exciting time. I think the kind of obvious follow-on from that is, and this is open to Mark and Hayden as well, is the industry coping well? Are we meeting those benchmarks? Are we hitting those new higher standards, or are we struggling a little bit because of the complexity of everything all happening at once?
I think for me, and the complexity is being self-generated. We can look at Annex 1 and say the new advent of Annex 1 has brought through all of these changes. But if I started when Annex 1 was coming out, I did a presentation on what's new in Annex 1, and we delivered it, and it's great. It's a great platform. And as you start to look for what's new in Annex 1, it was really apparent for what's not changed in Annex 1. And there's actually in Section 9, which is the environmental monitoring one, there are only 2 new elements in Annex 1, Section 9 environmental monitoring. And they are the requirement to classify a clean room for microbial burden and the acceptance of the adoption of more rapid techniques. Everything else can tie itself back to the 2008 version. Most of that can tie itself back to the 2003 version. For those of you old enough, there's elements of the 1998 version in there. And that thread comes through. And so what I then did was bring out a presentation that said, or a webinar, I forget what it was back in the day, what's not changed and why there are common threads that have been in Annex 1 for the whole time. For instance, trending. Trending was in 03, and they've amplified trending to help understand how to do it. And the problem is more within industry, and Vanessa, broached it. We've put those burdens on ourselves, and the regulators are looking to us to understand the regulations, because although they're there to enforce them, nobody's specifically training them in their interpretation. So they're looking to industry for how are they being interpreted. We're interpreting them to the nth degree. The sky is falling, not necessarily the sky is falling, but there's this new burden that's being applied to us, and it's not. It's a continuation of what they've always been saying with added clarity. I mean, one of the, for instance, is section 414. It talks about the pressure cascade. And now with gloveless isolators, there's a grade A to a grade C, and there's interlocks between the two, small airlocks, if you will, to get the bags, nested tubs from the outside in. Is that a grade B or is it a grade A? Is it B plus? So do I need 10 pascals and then 10 pascals to bridge? And so now we're applying all of these subtle rules, and the regulators are trying to keep up, and they will always follow the most common denominator, and that is what's hardest, not what's easiest. And so we're sort of like dragging ourselves, circling the drain. I heard mentioned once in a conference a couple of weeks ago of difficulty. We're making it harder for ourselves. And this goes back to your first question, is kind of what's wrong or what's the hardest thing that you face in industry. And it's that fundamental understanding, the reliance on experience that Vanessa, myself, and Hayden can bring and support, and just the application, because the regulation shouldn't be blamed for things getting harder. The regulation is following the trend of industry.
Excellent stuff. So I just want to move on a little bit now to technology, which has already come up once, and I suspect will be coming. They'll be coming up quite a lot. So, Mark, environmental monitoring programs have traditionally been fairly reactive. You sample, you wait for the results, you investigate deviations. How much is that model changing? Is that model changing at all? And are we genuinely moving toward kind of more continuous real-time monitoring? If so, what's kind of enabling it? Or is that just, not where the industry is at the moment?
No, as suppliers, we're being driven to put those tools in end users' hands. And some of that goes back to, we don't have the time to interpret the data in a meaningful way. And what does it actually mean about our confidence of control over finished product? So it is changing slowly. And again, I think the title of the RMM one, the Rapid Micro one this morning, was, sort of why is it changing so slowly? And there's this hesitancy to adopt new technologies, because I've got to stop what I've been doing for the last 20 years and adopt this new methodology, this new paradigm. And so we're trying to subtly sneak the technologies in such that you can keep doing what you were doing, go out, sample, test, report, analyze in a very formatted way, and then sneak in these little elements of continuous data. So the continuous sampling in a grade A for particles at 0.5 and 5, which was written into the 2003 version and implied in the 1998 version. So it's not new, but then we've got all of this data. So now I've got even more data coming in. Not what do I do? And that's where we're looking at tools to start an analysis, multivariate analysis, AI, kind of it's not full AI, but it's starting to bridge into that AI realm, where if you look at your particle count data and your microbial data and differential pressure and temperature, humidity and airflow velocity and gowning and all of those other elements, and what we're now trying to bring in is the visual inspection testing, because we're putting automated lines on visual inspection. So if that can tie into, which is finished product, if that can start to tie into the environment in this multivariate analysis world, in this AI world, and we can apply some rules to it such that on the back end of a media fill, on the back end of an aseptic processing simulation, then we can start to think about putting a dashboard in front of our users such that they don't have to analyze data in real time. They're getting this green, amber, red, your risk is elevated, almost a dial that says your risk is okay, it's low at the moment, it's getting higher because certain facets, line speed, depyrogenation, tunnel speed, carousel speed, number of nests, all of those other elements can start to bridge together to deliver the analysis that they need. But the reason we're having to do that is because there's been this drain from industry of experience that's able to make that interpretation. So we're building it in, we're putting that instrumental intelligence into the instrumentation to offset it leaving the industry. And now there's not the people making that determination, so they're going to the lowest common denominator, which leads back to the last question.
Hayden, do you share Mark's perspective, or is your take different?
No, I agree with Mark when you mentioned that, the shift to having continuous monitoring is real, but I also believe that it's a little still incomplete. We have the technology for continuous monitoring now, a lot of good offerings, but most operations are layering it onto legacy reactive systems rather than, I would say, fundamentally redesigning around it. And Mark touched on the hesitancy. There's a lot of good technology out there for continuous monitoring, but people don't want to disrupt the status quo, and they've been doing what they do for so long, and they have hesitancy around changing it, even though it might make their operations a little more efficient and have that continuous monitoring, to have that data in all one place. We still see that hesitancy, but I do believe that the shift is going to trend upwards in that direction in the coming years. And I would say that real-time data only matters if you're empowered to respond in real time, and that will require a process change. Many facilities haven't made yet.
I think that's a really, really good point, because I think, as I've said a few times today, putting the technology in is only a small part of the story. You have to do a load of other things around training, culture, process changes, etc. And getting that right is really hard in that shift. So Vanessa, have you got anything to add on that one as well?
Yes, I'm waiting patiently for my turn. So I have a couple points. First point is, I think that UNX1 is very clear that continuous monitoring is the expectation. Okay. And for both viable, active air, and non-viable. If you don't take that from the same document that I read, then your mind is biased towards your own needs and your site's needs and not that of the product or the process. Okay, so that's point number one. Even if you do continuous active air, viable and non-viable monitoring, it's still such a small percent of the overall air that is exposed in the critical environment. So absolutely you need to do that, because we need to optimize our best chance of getting the potential contamination that's happening while we're making the product, okay? And I'm talking obviously about sterile filled products. So that's the first point. The second point is, if you're part of a mature organization and you can manage other technologies, they're out there for real-time, viable and non-viable biofluorescent particle counting. Okay, there are technologies on the market. There are multiple suppliers who offer them, and they're really giving us, an insight real time and the ability to manage and be able to detect and perhaps quarantine certain portions of the batch that may not be provided under the same high quality environmental conditions. So, I think I've been in the industry. I was saying this yesterday. The number of years I've been in industry can now order a glass of wine. So I think that earns me the right to say that absolutely we should be doing continuous active air. I mean, this just should not be a conversation any longer. That's the bare minimum environmental monitoring you should be doing when we're manufacturing sterile products. And if you can, there is technology on there. We should be doing even better. We should be trying to get real-time understanding of the quality, right, in these environments. And there's technology on the market for that. So, I always encourage people to explore if biofluorescent particle counting works for you. It works in water. It's just such a better method for us to detect real time what's going on. And then if you can't, what Mark described is fantastic. It's it's disheartening that you had to do that to compensate for a lack of expertise and experience in interpreting the data, but also it could help speed it up because I think that while there's a numbers game, we don't have enough experts to interpret the data as fast as we need it to comply with the expectations. Those types of systems can supplement and perhaps present the data to the expert, and then they can make sort of that contextual understanding of how it applies to the process controls. So I think there's a lot of exciting things in EM, and I'm hoping that we shift to sort of this, model of both reactive and in real time understanding of environmental control.
I think, yeah, I was going to move at this point to talk about kind of the really hard problems, but I think we've run into some of the really hard problems already. But just to turn to you for a second, Hayden, I think one of the kind of contamination risk, which often doesn't get enough attention, is that kind of post-maintenance or post-shutdown restart period, when systems that have been idle or disturbed are brought back online. What are the kind of specific risks that you see that manufacturers need to account for during those kind of restart periods? And what does a kind of robust restart protocol procedure actually look like?
Yeah, so I want to focus here a little bit on a risk-based approach here, specifically for when we're looking at shutdown and startup protocols. And my specialty and forte is endotoxin testing, and this is a big concern with startups after a prolonged shutdown. So especially when we come out of that shutdown or a planned maintenance, this is where we have the elevated risk of endotoxin contamination, especially in water systems where that stagnation can promote gram-negative biofilms, which we all know are no good. So I want to get into a little bit about the risk-based phase approach for that. So I would say for phase one, or the first thing that is recommended is immediate post maintenance or shutdown is to sample all critical WFI or purified water points, point of use. Also storage tanks, and also worst case locations that you come up with in your facility that are potential spots for introduction of contaminants. So I would say once we identify those, use a 3-tier sampling approach daily for the first 3 to 7 days, or until trending shows control. Then you can start to reduce that. You want to test for endotoxin to the target of your WFI alert limit, which is less than 0.25 endotoxin units, alongside microbial counts, and also, if possible, conduct TOC or conductivity testing. If you're looking at once the water points of use are all tested and cleaned and all are trending in the right direction and look good, you can start looking at protocol specifics for your products and samples or in-process samples that are going to be using those same water tanks or the same CIP skids, stuff like that your product goes through. So I would say perform some sample method suitability testing on those, and look for any trends in your positive control recoveries, if those are elevated or showing low, or if you're actually having any potential introduction of endotoxin in those water tanks or in your skids or in your facility. But you don't want those in your sample, of course, so you want to be very proactive about that. And then once you're restarting your controls and your processes, I would say frequently and do intensive monitoring. So at least sample at least daily of your representative, in worst case, point of use until 3 consecutive days meet your specifications and your trends are stable. And then we can look at transitioning to, routine weekly or whatever risk assessment you have that says you have to test those point of use per frequently on your frequent basis. And then for, I would say, production resumption, you want to require endotoxin testing clearance on all of your critical utilities and process samples before releasing those equipment or your water lines. I think this approach can help definitely catch hidden endotoxin persistence early, even when those viable counts look acceptable. And you can provide data to update your control strategy and see where you need to do some more monitoring or you need to update testing at certain points of use.
So where you've got that kind of disconnect between standard microbial counts and endotoxin picture, how do you kind of create a contamination control strategy that accounts for that disconnect? I'm going to give that one to you again, Hayden, but I'm going to follow on with Vanessa because I think she'll have some interesting views on this as well.
Yeah. So can you repeat the question again? Sorry.
Sorry. So we touched on kind of times when you've got facilities where standard microbial counts kind of look perfectly fine, but the endotoxin picture looks different, sometimes very different. How do you how should a contamination control strategy account for that disconnect? What kind of approaches should you take?
Yeah, I would say look at it at a few different angles. You want to have specifically for endotoxin testing, and also I would touch on bioburden testing here as well. If you have some kind of disconnect between your results that you're having with like your endotoxin testing, and also I would touch on your alternative micro methods. So when you're building a monitoring control strategy, assessing these micro methods and understanding that disconnect, you really want to select those that best address your risks. So your rapid method that can also detect any organisms that you might not find with your traditional growth method. And you want to compare those two against one another. You want to make sure those plate counts you get on your agar plates are going to correlate to whatever rapid micro method you get. That way, your control strategy kind of looks at both angles. Is my rapid micro method really detecting what we might see on a traditional agar plate when we do our traditional bioburden testing? So I think performing those side by side, especially for bioburden, is essential for looking at your contamination control strategy on that end.
So Vanessa, just to follow on from that, from a kind of contamination control strategy design perspective, how do you build in the right kind of controls for a risk that maybe standard monitoring can actually just miss?
Yeah, I think so. When I hear discussions around endotoxin, this has sort of two ways endotoxin could introduce post shutdown. One would be the utility water system, right? And depending on how you do the shutdown and I've seen everything under the sun from complete water shutdown programs and then more controlled shutdowns. Sometimes after this type of water system impact or shutdown, if you have periods of either low flow of water or no flow of water, gram-negative bacteria can proliferate, right? There are vulnerabilities in shutdowns if they're weeks or months old. even if sanitization could kill those organisms prior to startup, the endotoxin could actually still remain. You may initially not see that when you release the utility back into service, and microbial monitoring could miss it. And then you could start to see symptoms of it if the biofilm was somehow disrupted once you got the system back into place. And it's important that you understand the system, the age of the system, where you're sampling on that utility system, and that you follow it up perhaps by a heightened monitoring program initially after you come out of a controlled shutdown. So that's one point. The other point is sometimes shutdowns happen at the end of the year, and depending on the climate, things happen to your facility. And because you haven't been walking the floor, you may have not noticed a new leak. You may have not noticed that an old leak that finally penetrated into control classified areas. And you may have not noticed things that just, maybe water was left there prior to exiting for the holidays or for shutdown. So I do encourage people not to have such extreme shutdowns, to sort of go into controlled shutdowns. You see in mature organizations, they'll have walkthroughs even though they're in shutdown, just to make sure. And I think this also depends on the age of your facility, the way that your facility was designed. It depends on, the relationship of the utility system to the classified areas. There's a lot of different ways in which endotoxin can show up in your product or your process. And you want to make sure that coming out of the shutdown, you're doing the appropriate cleaning, that you're doing the appropriate walkthroughs, that you're assessing for things that you may not do routinely, like water leaks, like looking behind mechanical spaces, looking into things that you don't do in your routine, perhaps daily walks of the clean room. So I hope that addresses your question coming out of a
Absolutely.
shutdown
Yeah.
for sure.
Yeah.
Yeah.
And I
I
think
would
that
like to
sorry, dive
add
in.
on. I would just say, like in a control strategy, when you're addressing your risk assessments, I think they should explicitly map out where gram-negative sources, like in your water systems, raw materials, equipment surfaces, and persistent mechanisms like biofilms, where those might be. And then you can use tools like a failure mode analysis or failure mode to identify points where those microbial kill doesn't equal endotoxin removal, and then focus on what could, like Vanessa was saying, what we could do to remove those gram-negative biofilms that are very indeed tough to get rid of, like such as things like hot withy circulation, turbulent flow, steam, hot steam, and chemical, processes like that will definitely help bring those endotoxin count down, especially after planned shutdowns.
And and I kind of add from a CCS perspective here that those maintenance shutdowns aren't always planned. And here in Florida, we have plenty of brownouts where various systems will just burn off. And here we are coming up to July, so we're looking at hurricane season too. And if a hurricane goes past the coast, and certainly if it comes on shore, it'll change the flora and fauna of the water, the base water that will be purified for PW before it even gets GWFI. And so that whole shift of the microbiome will change on you as well from outside in. So having a robust tested plan, and I think that's where the CCS will come in, is you have to don't just trust fingers crossed in the systems, test them, challenge them, make sure that your scheduled and your planned maintenance that takes place can be done in a hurry, and so it can be done reactively. So yeah, test to make sure that test and verify.
I think that's a great point. I can probably close this topic that yes, Mark, test, 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 but coming out of a shutdown, you need to have checks and balances in your system, bringing it back on. So sometimes going into a shutdown rocks the boat in a way that you didn't predict. And I'm talking about gaskets that go unchecked or piping that may have had a dead leg and it was already vulnerable, but now it's really showing and there's an issue or gram negatives were allowed to proliferate. So, it's kind of like that old water heater you've got in your house and you're hoping you get one more year out of it so you don't have to replace it and spend $15,000. The same concept applies when you're coming out of a shutdown, that testing is fantastic and it will confirm the controls you have in place. But you have to have strong controls coming out of a utility shutdown or a maintenance shutdown, making sure that you're checking all aspects. You don't just turn it on, let it run, and hope for the best because your testing is good. So you need to have, that's where the CCS, like Mark said and Hayden said, helps you design the controls and make sure coming out of a shutdown, you're checking gaskets, you're doing the maintenance, you're replacing things, you're opening pipes. If you've got a flush, it's one thing I've learned about water systems, flush and sanitize coming out of shutdowns. That's what you need to do. So,
Yeah, and in the semiconductor, it's even, it's zero point 0.1 micron. It's nanometers, not just, 10 and 25 microns that we're looking for. Clean water is quite different.
Mm, mm. So I want to pile on more challenges at this point. So in the session agenda, it refers to kind of building capacity to detect challenging contaminants. Mark, just turn to you for this one for now. What does challenging really mean for the industry at the moment in practice? So what are the categories of contaminant that current monitoring programs most routinely underestimate or occasionally miss entirely? And what capabilities can we use to kind of close those gaps? What does it require?
Um, Again, there was again. There's a reliance on we've always done it that way, and therefore it must be good. We've always done, for instance, settle plates for the last 25 years. We've used settle plates as a mechanism to do our continuous monitoring because we'll just swap plates out back to back. And so there's this reliance on the way we've always done it that we're not looking at potentially new contamination. So a lot of the new technologies tends to be, we're looking at gaseous emissions in freeze dryers. So where we start to look at freeze dryer technologies, they're not allowed to leak or leach any chemistries into that area. So lyophilizers have a new set of boundaries. When we're bringing a new facility online, there was an old paper, crumbs, about 15, 20 years ago that looked at the outgassing of the glues that you're using in vinyls, the adhesives that are being used to hold the Tyvek to the lids of the tubs. And when they're exposed, they're going to outgas. And are those gases causing a chemical change? And especially now we're looking at, large particle, large entity biologics where they're susceptible to those changes, to chemical changes, not just particulates. Also, from a particulate perspective, if we're getting smaller particles in air, so 0.3s and 0.1s, they're acting as nucleation points for a lot of the proteins that are being filled, and we're not concerned about anything smaller than 0.5, where those biologics are exposed. And a lot of those are exposed in smaller facilities that are using either glove boxes or hoods. So there's a higher interaction with operators and personnel. So there's a broader spectrum of the particulates. So right now we're sort of reliant on the way we've always done it. And if we were to look for what else do we need to be aware of, well, there's the chemistries in air that might affect the quality of product. And there are smaller particles that might affect the efficacy of some of the newer biologics. The instrumentation for those all exist. We've just not adopted them because now it's more data and now I've got everything else to worry about. And so until they regulate against it, we're not going to do it. But as an industry, are we monitoring them as side projects that might yield additional information that creates a better quality product at the end?
Vanessa, I saw you smile at the mention of settle plates there.
I don't know. I have a lot of strong feelings against settle plates because I feel that they are very incapable of detecting contamination in the areas in which they're used.
You're in good
Um,
company, Vanessa.
yeah, like I said earlier, I very strongly interpret based on my experience, but also understanding of microbiology that the Annex 1 requires continuous active air monitoring. This is not only like a regulator telling us to do that, but it just makes good sense for the product and the environmental control, right? And as a scientist, I want the data to be meaningful, and I just really feel using a settle plate under those circumstances for, really high velocity air, understanding what we know about airflow, visualization, and even all the computational fluid dynamics that are coming out. They're just showing it's just whizzing past that settle plate. It's not actually accumulating. So I really feel strongly against the use of settle plates in critical environments. That is been my consistent position throughout the industry for quite some time. I also think when we talk, Ian, about challenges in the industry, I'm a hardcore, traditionally, classically trained microbiologist. I grew up with agar. I love agar. I love the smell of knowing agar is cooking and autoclaving. Um, but I also am a very practical, science-based person, and I know that which only we can grow on certain agars is what we detect. And I
Mhm.
love the shift towards alternative rapid micromethods that are not growth-based, that could tell us the presence of organisms, whether or not they're alive, whether or not we can grow them in agar. And the scientist inside of me, the logical person inside of me is like, yes, that's exactly where we need to go. But I think we need to be selective in which we use those. They need to make sense. The technology needs to match the ask, right? And it needs to be developed in your CCS. So where in your product and process and your environmental controls could you use an alternative rapid micro method? And what's the value you're gaining from that? And how does it tell you, improved controls about your product? So I think that it's exciting that we are on sort of this precipice. But if you talk to folks like Dr. Michael Miller, he's been trying to push for rapid micro methods for 30 years. And I've been trying to push for no saddle plates for 20 years. And so, the adoption of new technologies, of understanding what's present in the air or in the sample or in the water, really is on us to develop and to adopt into the contamination control strategy. And I hope conversations like this help push us towards that, because the scientist inside of me is like, absolutely. How can we not? If I can do better and I have methods that can support that, that's what I need to be doing. And I think the Annex 1 clearly supports that position as well.
And I
In,
think what
in,
you're saying comes back to the point that Michael made about, we tend to have we've always done it that way attitude for a lot of things. And, you're kind of similar in the sense that you like the particular smell of alga. It's like you can't avoid it. We're human beings. We like doing things the same way again and again. Hayden, have you got anything that you want to focus on in terms of that? How do you get people beyond the point of we've always done it this way? What do you see from the industry?
Yeah, I think we're headed there. People seem more open to the idea of new technologies that's not only gonna, lessen the burden of the personnel in the lab and in the facility, but also, give them good data they can stand behind to make informed decisions about their processes and their results. Um, so I think, I know people we touched on this earlier, but it's the last, I would say, 3 to 5 years, there's been an emerging trend of new technology that would really benefit a lot of people. But I think there is still, like we touched on, there's this hesitancy that, like we said, we've always done it this way. It works. Why change? And people don't want to do that. But I think we have to just look at the bigger picture and see, what what these new technologies can really do for us in 10 to 15 years, even from now, and how much more processes we can get done and how much more product that people need we can get out to the market in quicker time. So I think we'll still see some challenges just with facing the workload of switching and people having that hesitancy. And the regulators are going to be really, I think, pressing down on folks to look at making these changes, and I think they will eventually come. I think it's just going to take some time still, and we need to have good data behind it to make those decisions and to say that what we're doing now against what we've done in the last 20 years is actually working. It lines up, and that data is actually correlating with what we see and what we saw in the past.
You mentioned, sorry, you mentioned data there, and I guess like we've said, what we were talking about at the start, it's very, very difficult if you haven't got the high quality of data that you want to start with to actually get a really good outcome from it. And when we move to, building more resilient and proactive CCS, you have to actually, the data is really important in that context. The other thing is that you have different data types, and creating kind of trend analysis across different data types can be quite difficult. So, Hayden, maybe you want to touch on this one. How do you perform kind of meaningful trend analysis when your contamination data is expressed in fundamentally different units across different methods, and what kind of approaches allow you to monitor contamination trends holistically?
Yeah. So when we are when we're looking at meaningful trend analysis with our contamination control strategy, we want to perform this meaningful trend analysis by focusing on patterns and shifts, excursions, and even risk signals across methods, rather than expecting perfect alignment between different reporting units. So as we know, CFUs, our traditional aggregate plates, colony forming units are going to be different than what rapid micro methods are going to be giving us. So those are typically in relative fluorescence units, biotics, either a pass fail, depending on which detection method you're going with. So these RMNs and traditional CFU capture overlapping, but I would say distinct aspects of contamination. For example, a rapid micro method can often detect a slower growing organism or a stress organism faster than an aggro plate. So one organism might take 5 days to grow on an agar plate, but maybe using a rapid micro method, you could pick that up in 24 hours. So that would definitely strengthen your contamination control holistically, I would say. And some key reconciliation approaches is, I think I touched on this earlier, but if you are looking at going from your traditional bioburden filtration method to a rapid micro method, we want to make sure that we're establishing a strong correlation to plate counts through validation testing and side-by-side testing with environmental isolates. I've seen many RMNs show good correlation supporting conversion factors or equivalent CFD mapping. We also want to overlay trends in a unified contamination control strategy, like dashboard. I would say dashboard for cross comparison while maintaining those separate results for context, and also run periodic parallel testing to verify ongoing correlation and treat excursions holistically for any kappa or continuous improvement of the methods.
I think, yeah, it's a really interesting topic. I want to move on a little bit to talk about proactivity, because everybody, proactive is a huge buzzword, not just in pharma, across every single industry where quality control is super important. And my first degree is in philosophy, very long time ago. But the first thing they tell you in philosophy is you've got to define your terms. So, Vanessa, maybe you can take this one. So, proactive contamination control, We all want to do it. It's an aspiration. We use the phrase all the time. What does a genuinely proactive CCS actually look like in practice?
Yeah, I think it's a great question. And I'm happy that we're able to have these discussions. So I think just inherent to the way that we are thinking about what is proactive and what is actually controlling, right? And then what is the data that we're capturing to measure the effectiveness? I think a proactive contamination control strategy has a lot of different elements. First and foremost of which is you articulating the actual understanding of your process, of your technology capability, and right sizing the controls for what you need them to be. Okay. If you talk about, a lot of times, or at least historically, contamination control has been taking bioburden or taking environmental monitoring or particulate or all these different measures to say, are we doing what we said we're doing? And does the data show that? That's not controlling anything. That's detecting whether or not your controls are working. So then you've got to walk it back upstream. What are the controls you have in place? And are the controls appropriate for, whatever program or system you're trying to control? And how do you design that? So considering your facility, how it's zoned, how it's classified, what are the right size controls that you have for your facility, not overdoing it? Thinking about how you clean the facility. What are the types of agents that you have, and are those appropriate? Are the frequencies actually appropriate, right? And this is what I talked about earlier. Shifting the mentality from compliance perspective. Yes, I have a cleaning and disinfection program. Yes, I rotate my disinfectants. And yes, I do a rinse, right? But how do you do that? And which agents are you using? And are they working? Are they overworking? Can you scale back so you have less harm to the facility and therefore you have less rust or wear and tear, thus creating a different microbial problem? So it's really about understanding every contamination control strategy that I've seen that is brilliant describes the process and the controls that they need that are appropriate for that product, that process facility. And that is, first of all, where you're proactive. If you've got fundamental facility issues or design issues, you need to correct them. We can't just collect EM samples until they show up with better data. We need to correct facility design issues within reason, right? Because there was a question earlier about aging facilities, right? I I don't have the money and I need to do production, so I can't just magically appear a brand new facility according to current today clean room design standards. But are there changes, very meaningful changes you can make to the facility that will improve proactively and prevent contamination, right? Maybe it's changing the material for your floor. Maybe it's changing where the joints meet the wall and the floor, right? Maybe it's upgrading that. Maybe it's putting certain stainless steel blockers to prevent the walls from getting nicked, exposing materials and growing contamination. So understanding your facility, understanding its flaws, and implementing the controls that are appropriate for your facility. Not just because Annex 1 tells you to do a control, but how does that control make changes for your facility? So I think the more that you can understand your process, the more that you can demonstrate you're implementing controls that are appropriate for you, for your facility, those are proactive, truly more mature contamination control strategies that I've seen. And then you build in the detection capability to say, yeah, actually, the control I put in place is working, right? Or it's not. If it's not working, that's just as important as it is working because then you can change the control. And then you have true life cycle management of your CCS. So whether or not something's working should feed back upstream and you should change the control. And that's the way that we can get towards a proactive model. Now, I also see people just scrambling to put together their CCS in sort of a more, you know, words on a paper type of way. And that can be a good start. If you don't have, a grip on all the controls you have in place, start by listing out the systems and then start by marrying them with the controls that you do have and determine whether or not those controls are appropriate, right? And a lot of people are doing risk analysis through like a HACCP type of model, which is fantastic because it asks you questions that maybe you've never interrogated to your process before. So where is the risk happening? What types of contamination could happen with that risk? And what could go wrong if you fail to mitigate that risk? And so asking sort of the next level questions helps you get back into that proactive mentality. And I think that that could be really important as we go forth and mature the way that we do CCS.
So before we move on to questions from the audience, and we've got some really good ones, I just want to touch on one last question for all of you. And it's this. If you could change one thing, only one thing, about the way the pharmaceutical industry approaches contamination control, not specific technology or regulation, but a mindset, a habit, an assumption that we make as organizations, what would it be? And I think let's kick off with Hayden.
Yeah, and mine is be a little funny, but I do believe in the proactive approach. With Vanessa just touched on a lot of good points. So if I could change one mindset in how we do approach contamination control, it would be, shifting from that reactive testing, saying, oh, pass the test, pass the make it, to proactive design first prevention embedded in, their everyday habits of their processes and their contamination and control. I would say too often we treat contamination control as something the lab or QC catches at the end rather than a core possibility, or core responsibility, I should say, built into facility design, process decisions, and daily operations from day one. A stronger prevention mindset, I would say, where every team member thinks, how can I eliminate the risk upstream? Would I think would reduce reliance on monitoring alone and truly strengthen the CCS for a better outcome.
Vanessa, over to you for this one.
Yeah, so I think that there are a lot of ways that we can change the mentality. I think we should change it from being a checkbox activity to having real conversations. I think we should change it from just looking at data, but providing context to that data and perhaps, having conversations between microbiologists and engineers and process experts to say, okay, I may need to have a conversation with you to understand the process a bit better than I know it, so that then I can tell you where those risks are from a contamination perspective. And so I think opening the conversation, making sure that it's not just having a CCS and keeping it moving, that you really embrace why contamination control is important and how, from a business mentality, it can also help you reduce unnecessary burden on investigations, et cetera. So there's also a business perspective for why contamination control just makes sense.
And finally, Mark.
I'm going to echo what Vanessa said. It's get out of the swim lane. When we look at quality issues, we sit in a cleaning. Okay, we've got a swim lane for cleaning and cleaning validation. Our environmental monitoring, our 0.5s, our 5s, our CFU, our whatever it might be. Everybody just lives in their swim lane. And the only real way of getting out of that is to start having the interdepartmental or the intergroup mindsets which the CCS tries to bring. Okay, that says everybody should be collaborative in trying to, because contamination doesn't live in a swim lane. Contamination lives across the facility, and it will bridge and it will jump, and biofilms will go wherever they want anyway. So it's to get out of that swim lane mentality, and the only way that we can help them do that is to give them the tools to reduce that burden of data so they can start making decisions and to start looking at the interpretation of that data and how it applies. So it's on industry, and it's on us to try and put the tools in their hands for them to take them and put them into practice, but get out of the swim lane that they live in.
Great stuff. And let's cover off a few of the questions from the audience. And there was a couple here, which I'm kind of going to combine into one around how you deal with aging facilities, which I think is going to be top of a lot of people's list of questions. So what are some of the components of CCS that you can foresee would be commonly difficult if you have aging facilities and you need to meet the requirements of Annex 1?
I can take that. I think, can I weave that, Ian, with the
Yeah,
other question on unidirectional flow path and
of
bidirectional
course.
flow path? Because this is a very valid facility aging question. So first, I think that we should be honest on whether or not there are portions of our material, product, process, waste flow path that need to be absolutely unidirectional, right? We absolutely cannot have product and waste by any way bidirectional, right? And assuming that that fundamental idea has already been integrated into your facility, perhaps we're talking about bidirectionality when it comes to people and materials, okay? And there are controls that you can put away, put into your contamination control strategy that will help you mitigate that. So first, I would recommend taking a look at where those bidirectional flow paths happen and have an understanding or a description in your CCS that says what's going on and what are the risks associated with that, right? And then if you absolutely cannot reconstruct the facility to make something unidirectional if it needs to be, are there temporal controls that you can put in place so that people and material are not in the same area at the same time? And maybe it's only certain material, clean, single-use systems, certain tanks or something like that that need to come through. You don't have people there at the same time.
Mhm.
If, for example, you have both bidirectional people and material, maybe you need to clean the area if you've got people dressed a certain way, right? And you need to bring in clean materials. So maybe in between that, you can clean. There are solutions to a lot of the aging facility problems, whether or not we want to implement those solutions and the resources needed to do that is a different question. And you have to, explore what's feasible for your facility. The other thing is some of the inherent unidirectional flow path requirements are so that we don't have people doing on one side of a room a process for a differently classified area of the same room, and then people dressed in a cleaner process. And that there's, fundamentally from a contamination control perspective, that just doesn't make sense, right? If
Mm.
I'm in street clothes and you're gowned, to grade C or D or whatever, or if I'm gowned, with hair exposed and stuff like that, but you're in a sterile garment or a sterilized garment, then that doesn't make sense too. So preventing some of the processes or transfer processes that happen with bidirectional needs to be better controlled, and you should describe that in your CCS. So there's a lot of ways in which we can adapt to some of the issues we experience with aging facilities. If and when none of that is possible, then we need to think about sharing rooms, reallocating, square footage or the space in the room, and really have the harder conversations during those shutdowns. Could we do some construction? How could we change one room, maybe move the doors, right? Maybe create a pass-through, a smaller pass-through to control the materials going in. There's a lot of different ideas. I think you have to show that you've explored them and that you understand the risks associated with each of the different flow paths and that you've got controls to help you bring that contamination risk down. That's really the point. Hopefully that makes it clear.
Excellent. Time for a couple more. I think this one might be my fault because at some point I was talking about change and how people are sometimes resistant to and have fear of change. And one of our commenters has given a really long comment on this, but boiling it down to its question, as a scientist, I'm actually quite open to and excited by rapid and advanced EM methods. However, it's the unknown consequences and uncertainties associated with those approaches that often make one a bit hesitant. So I guess the question really is kind of how do you get over that? How do you get through that as a scientist? Is it through more awareness that that's the way you're thinking, or is it something methodological? How do you do it? Maybe Mark, do you want to pick that one up?
Certainly, I think from a rapid environmental monitoring perspective, it I think what we've tried to do with that technology is use it independently. I'm going to use my RMM, use that autofluorescence data, and how do I replace everything else with just that singular piece of fluorescence information? And that, I don't think it was intended that way, or if it does, that's where it's ended up. Because it's really useful supplemental information. It's additional information. I can look at my point 0.5 and my 5, and there's a relationship between those two. Again, let's not swim lane the data. Let's not put RMM and replace everything else, because I don't think that maybe through data, there's one instance once of an RMM and a CFU being captured at the same time. Now, if you've got an environment where there's less than one, typically anyway, it's never going to appear two places at the same time. If it does, then you've got a much broader contamination issue to worry about.
Mm.
So it's how does it fit with what I've got rather than how does it replace what I've got, might be an easier way of seeing where that science lays itself within your overall picture, this holistic look at the facility and not the swim lane look at the facility. How does it bolster my microbial data? How does it bolster the settle plate, for instance? And as much as we deride them, they have a place.
Mm.
And so there's a risk analysis associated with the science. Do you monitor the first air? Do you monitor the process? Because microbes existing in first air shouldn't be there. It shouldn't be able to get through the filter the same way particles do. But it being encapsulated within the process, that's where you want to be monitoring the microbial activity to ensure it either doesn't recirculate, fix it. If there's a risk that identifies a problem, then it's there to mitigate, not there to just measure. So it's where does that science add the value, and that value becomes a commercial issue as much as product quality. So I think scientifically it's trying to integrate it and not to use it as a replacement tool.
Absolutely. We got one last question.
Ian,
Brief
can I
answer for this
add?
one. Oh yeah, go
Can I
for it. Go
just
for it.
quickly add something for
Mm.
the hesitation, the fear, the unknown consequences and uncertainties? I would just want to say, like, you're not alone. You
Yeah.
don't need to do this alone. There are brilliant studies that are published on a lot of the new technologies. All of the suppliers, all of them, and I've studied all of them in pharmaceutical microbiology, have primary validation data. That's the expectation. If you listen to the TR33 and all the recent updates, number one, suppliers should have primary validation to prove that their technology works. They do. All of these new arms have it. I think that you communicate with others. So if you've got a specific system you're interested, contact the supplier. If you can't figure that out, we've got most manufacturers in the world implementing all the new arms in the world, and they've got regulatory filings and they've got regulatory approvals. It is not clean. It is not easy. There will be hurdles, but we can do this collectively as a group. We can do this. And I think that the value of arms, the value of new data analysis, like the multivariant that Mark mentioned, the value of doing better is for the product. And that's on us to take that leap, to push through those fears and lean on all of our colleagues, myself included. If you've got questions about, I can put you in touch with the right people who have done it, who have published the studies, and who have regulatory filings approving it. So we need to push through some of those fears. They're very valid, but we can do this. So I just wanted to share that, like, all the arms out there have been really well established, and we should be thinking about how we can get them in there as soon as possible. And the business case for the larger leadership, it's there. The value having this detection and capability of all these bioburden analysis and air sampling systems, it's there for the bottom line for the product quality. So we've got to get better as a group of microbiologists, and understanding the business needs, pushing beyond those fears. And I think we can do it.
I think that's a perfect place to end it. And interesting, you mentioned about the business case, because we have a session on day 3 that looks specifically around how to build business cases that actually persuade the C-suite that you've got to get this stuff in. So perfect ending for that one. So thank you very, very much to my panelists, Mark, Vanessa, Hayden. You've all been absolutely fantastic. You've probably seen in the chat, we've had some great feedback about the panel overall, which is lovely to see. A quick word and thank you to the audience, but a quick word and a thank you to our sponsors, so Veolia and Particle Measuring Systems, without whom none of this would be possible. So please do check them out, connect with them on Swapcard, etc. That's it for today. This is our last session, and it's been an absolutely wild ride. I've been in every session so far. I will be in every one tomorrow and every one on Thursday as well. So join me again at 12 o'clock tomorrow afternoon, UK time, and I hope to see you there. Thanks again to everybody.
Thank you. Thank you for having me.
Thank you so much.